Hey everybody, today we're going to be talking about osmosis. So we're going to be on page 10 in your packet. If you did not take the notes on diffusion yesterday, that is going to be covering this kind of stuff. So if you did not see this stuff yesterday, please make sure that you listen to the diffusion notes before you work on osmosis. So on osmosis, so we're still talking about the movement of molecules through our cell membrane. Remember, a cell membrane is semi-permeable, meaning some things can move through. So osmosis is just the movement of water. So we said that water was small enough to be able to move through the semi-permeable membrane. Now, when we talk about osmosis, we are still dealing with passive transport. That means we're going from an area of high concentration to low concentration. So we're going high to low. Yesterday I mentioned it's like a slide. If I'm at the top of the slide, I'm up high, and then I'm sliding down to the low part of the slide. This requires no energy. If you want to add that to your paper, no energy, please do so. It's a good reminder. Now, the movement of water. So the water is going to keep moving until we reach those at equal concentration level. We're going to reach equilibrium. So equilibrium is just that it's equal on both sides. Now, if we look at the pictures below, the first picture is not in equilibrium. Okay, even though the water is equal, we're actually looking at the concentration levels. So here I have a few dots. Here I have a lot of red dots, but my dots can't move. So this is kind of like the Kool-Aid bottle. So here I have really dark, strong Kool-Aid. So in order for this to be equal, I need to add more water to this side. So if we look at the second picture, we have equal concentration levels now. So I lost a little bit of water on this side and it moved on over. So again, like the Kool-Aid bottles, if I had two bottles, I'm going to add a little bit more water to the other one to make the concentration levels equal. So here's just another picture of that concentration levels. So again, we have our selective permeable membrane. Here on the right-hand side, I have a higher concentration of red dots. Okay. Here I have a lower concentration of red dots. So I want these concentrations to be equal. So that means I have to add more water to this side. Okay. The dots do not move. All right, so our cells constantly go through osmosis, depending on the type of solution that they are in. So there's actually three solutions that we're going to write down. We're still on page 10. We're going to write down hypertonic solution, isotonic solution, and hypotonic solution. So please do that now. All right, go ahead, flip to page 11. So on page 11, we're going to talk about each of these solutions. So a hypertonic solution. This is a cell that is in a hypertonic solution. So what that means is that water is going to move out of the cell and the cell is going to shrink. So the reason that the water is moving out of the cell is because there is a higher concentration of, we'll say, dots on the outside. Okay, so the water is leaving the cell to try to make this whole thing equal. Now, I like to think about hypertonic solutions. So if someone is really hyper in my class, I'm probably going to kick them out. Okay, so that's what I think. I think hyper, I'm kicking them out. So we're kicking the water out in a hypertonic solution. Okay, because again, we're trying to equal the water, or we're trying to equal the concentration levels. So water moves out of the cell and the cell shrinks in size. Just like if I kick somebody out of the classroom, it's going to shrink in number. The second type is isotonic solution. Now on your paper it says equal parts solute and water. So solute is like that sugar or salt. So with isotonic solution, we have equal movement. So water is going to be moving in and out equally. Excuse me. So that means that there is an equal amount of dots on the inside and equal amount of dots on the outside, okay? And water is just flowing in and out. This is where we want our cells to be constantly, okay? So when this happens, there's no change in size. The last one is known, or excuse me, for isotonic solution, I think iso happy because we have equal movement and there's no change and we are happy that our cells are good. So iso happy that my cell is not changing size. The last one is a hypotonic solution. This is when water is going to move into our cell. So if water is moving into our cell, it's going to expand and get bigger. So that means we have more salt or we have more sugar on the inside, more of those red dots on the inside. So the water is rushing in, trying to equalize everything. So hypotonic solution, I think hypo, hippo. Okay, and if you want to write that on your page, hypo, hippo, because it's getting large, like a hippopotamus. It's expanding out. So when we talk about these three things, again, here we have our normal cells. This would be a isotonic, excuse me, an isotonic solution. This is not in your notes. This is just another picture. Hypotonic, it's going to expand, and sometimes our cells can actually burst. Or here we have our cells are going to shrink and shrivel up. This is a hypertonic solution. Again, hyper, the water is being kicked out of our cells. We see here, the water is being kicked out and our cells are shrinking. Hypo, the water is going in and they are getting ready to expand and burst. Here's another picture of what our actual cells look like, our red blood cells. So this would be a healthy red blood cell. This is a expanding red blood cell. So if it's expanding, it's going to be in a hypotonic solution because hypo-hippo, it's getting big. And this is a red blood cell that is in a hypertonic solution. Water is being kicked out of the cell, so it's shrinking down. All right, so again, this is not on your paper, but it's just a little example. So if we take a look here, there are more water molecules on the inside of the cell than on the outside. They will move from an area of higher concentration to lower concentration. So if we look here, all of my blue dots, and the blue dots represent salt in this picture. Now, salt sucks. All right, keep that in mind. Salt sucks. When you eat something very salty like pretzels or peanuts, you usually get really thirsty because the salt is drawing the water from your cells. So you become dehydrated. That's why salt sucks. So in here, we have just a little bit of salt. Outside of my cell, I have a lot of salt. So the water is going to leave my cells. So if it's leaving our cells, what kind of solution is that going to be? The water is leaving my cell. So it's going to be shrinking in size. This is going to be a hypertonic solution. Okay, so it's shrinking because again, the water is leaving. Another example, here I have a 10% salt solution on the outside and 20% on the inside. So now I have more salt on the inside of my cell. So the water is going to be rushing in. If the water is rushing in, it's going to now do what? It's going to expand. It's going to get larger. It's going to get very big, round, like a hippo. So if it's like a hippo getting larger, it's going to be a hypotonic solution. And the last one, I have 10% on the inside and 10% on the outside. It's very happy. It's equal amounts on both sides. So if it's equal, it's going to remain the same size. And the cell is just so happy. So if it's so happy, it is an isotonic solution because it's equal. Water can move in and out, equal parts on both sides. All right, so we are back in our packets now. So we are filling out these bubbles at the bottom of page 11. So in the top one, you're writing if a cell is in a solution that is, we're going to be going through all these solutions again. So in those bubbles, you're going to fill out hypertonic solution or just hypertonic, isotonic, and hypotonic. We're going to start with hypertonic. If we have more solute and less water, solute again, write yourself a reminder, put a little arrow right here. Solute is like salt or sugar. You want to do salt because salt sucks? Maybe that'll be a little bit easier for you to remember. Hypotonic is that there's more water and less solute. Isotonic, there's equal amounts of solute and water. So if I have less water, okay, that means that water is going to be flowing out of my cell. If I have more water, that means that more water is going to be flowing into my cell. And if I have equal, water will be flowing in and out equally. Again, if you need to pause this to write these down, please do so. So with a hypertonic solution, if water is flowing out of my cell, okay, to try to make things equal, we're kicking it out because it's hyper. Water is flowing out. That means my cell is going to shrink. In a hypotonic, hypo-hippo, if water is flowing into my cell, that means my cell is going to be getting bigger. It's going to expand out. And finally, if water is flowing in and out equally, the cell is going to remain the same size. All right, once you guys have that done, move on to page 12. You guys are going to take a look at these six cells. We have our red blood cell, and then we also have a plant cell. So take a look. Remember, the cell wall is the outer edge, and then on the inside is our cell membrane. So we're looking at what's happening to the cell membrane. So based on what you guys see, I want you to write down whether the cells are placed in a hypotonic solution, a hypertonic solution, or an isotonic solution. So go ahead and hit pause and take a guess as to what these three are in, and then what these three. So you'll use the word isotonic twice, hypotonic twice, and hypertonic twice. Again, hit pause, fill out your answers, and then see if you're right. If you're not, go ahead and make those corrections. All right, so you should have your answers down, and we're going to take a look at our red blood cell. So our red blood cell right here is looking pretty big. So this red blood cell is going to be in a hypotonic solution. Hypo-hippo, it got a lot bigger. This red blood cell is starting to shrink down. It's pulling in on itself. So if it's pulling in on itself, that means water has left and our cell is shrinking. So this will be a hypertonic solution. Hyper because we're kicking out hyper people. Hypertonic, it's kicking out the water. Our last one, our cell, it's a normal happy cell. So this would be an isotonic solution. Iso happy because my cell is perfect size. Looking at a plant cell. So in our first plant cell, we notice that our cell membrane has pulled in on all these sides right here. So if it's pulling in, that means it's shrinking down. The inside of that cell is a lot smaller. So this is going to be a hypertonic solution. The center, everything looks normal. The cell isn't getting bigger. It's not pulling in. This would be an isotonic solution. And then in our last one, we notice that it's starting to actually bulge out on the sides. This will be an hypotonic solution. Again, hypo-hippo. Make sure your answers are correct. And we are going to stop there for today. If you did not finish Model 3 from yesterday, please make sure that you do that now.